Hello, I'm here with Professor Bart De Strooper, group leader at the VIB-KU Leuven Center for Neuroscience, the Francis Crick Institute, former director of the UK Dementia Research Institute, and a recently minted commander of the Order of the British Empire. So, Professor Bart De Strooper, if you could just briefly introduce yourself.
Mm-hmm. I'm an investigator at the KU Leuven, doing research around Alzheimer's disease, and well, I'm quite senior. My career is now 40 years. That I try to understand the reason why our beautiful brain, at a certain moment, because of certain circumstances, starts to degenerate and causes dementia.
So, how would you describe the main focus of your current Alzheimer's and dementia research?
Well, we are at the moment in a real breakthrough phase. When we started, we didn't understand anything of this disease. We had just a picture of what happened in the brain, but that's a picture when people have died already, so it's a very late stage with the plaques, amyloid plaques and neuronal tangles and neurons which are gone, and etc. etc. And so at the moment we have a quite good idea about how it starts and how it ends in that situation. And so what I'm trying to do is now to get different steps which go from the very beginning where the brain is normal to very end where the brain is a catastrophic site where everything is broken and where the neurons are dead. But what are the steps? Because this process is about 20 to 25 years. So in the beginning there is a long phase where there is some damage in the brain where you can see the amyloid plaques, for instance. You can see some neuroinflammation, but the neurons remain healthy and remain functioning. And so that's really what I want to investigate, what's going on there, and what basically makes that after 10 years of such an equilibrium between amyloid, inflammation, neurons which compensate, what happens at a certain moment so that a system collapses and that we go in the next phase of Alzheimer's disease, which is basically the tau pathology.
Mm-hmm. And regarding the phase, you have coined the new cellular phase of Alzheimer's?
Yeah, that's the name I've given it 10 years ago. But in the meantime, we have a more sophisticated view on this cascade. So the cellular phase is when the different brain cells, microglia, neurons, astrocytes, but also the other cells, basically react to that amyloid plaque, and you get an inflammatory, destabilized environment. And that's what I call the cellular phase. But then you have the next phase, which is in fact the induction of the tau pathology, which is an intraneuronal in the neurons pathology, and which then basically provides a substratum for the next cascade, which is a cascade of cell death, neuronal dysfunction, etc. And then you have a last phase, because you can lose quite some neurons before anybody will see that you have a disease. So I think everybody would agree, if you are losing neurons, that it's not a good thing. But the brain is so plastic that it continues working. So the last phase, and that's what we call in the clinic Alzheimer's disease, is when you have massive neurons gone and that you start to affect the function of the brain. And that's what we call dementia. And so the cellular phase is only a part of this cascade.
For cell work, you and your lab use NAMs. So how would you say that the use of stem cells and 3D cells are being thoroughly used in your lab?
Yeah, well, it's a bit of a strange question, which I get regularly. Of course we use NAMs. We used that already when I started. But we moved from NAMs to mouse and animal experiments because the brain is so extremely complicated. There is no NAM for brain function. And so we use still both, and we have become much more sophisticated because there is of course difference between human and mouse. So you want also to have a human cell and a human environment to study the disease. And so what we are using a lot, we have tested certainly organoids, brain organoids. I found that fascinating, but it's absolutely not ready for Alzheimer research, whatever people claim. You cannot reproduce what we can reproduce partially in mouse, but we cannot reproduce it at all in these organoids. And so I also doubt that a lot of claims made in organoids are actually reflecting the real facts. But so we have tested that, and what we also use a lot is when we try to simplify our question, once we have a molecular insight. So the mouse we use for pathophysiology, so where you want to know what is the impact of something on a system, on a biological system. The NAMs, the cell culture, the iPSC cells, we use when we have a molecular biology question, where we want to know molecule A is influencing molecule B, and that you can much easier study in an in vitro model. And so we have really sophisticated, so organoids I've explained already, but we use now a lot of 3D cultures, 3D and 1D culture, which is basically 1D is a classical cell culture where you grow your cells on a flask, on a surface, and 3D is where you use a matrix and where the cells grow on top of each other and in neighborhood of each other and influence each other. And so that's the 3D culture. And we are trying to increase that and to use that in automatic systems, etc., for certain questions.
And you just mentioned the use that NAMs and for different questions. With the evolving technology of AI and also computational biology, how do you use these technologies to complement your research?
Well, we use this, of course. It's a fantastic tool, but it's like you use WordPerfect, like you use any other digital tool. It's, of course, an order of magnitude more useful, I must say, but it comes also with all limitations. So, I mean, I don't understand why AI is linked to NAMs. AI is used for everything, for writing papers, for making projects, for analyzing data, whether they come from NAM experiments or non-NAM, from animal experiments. All the experiments are analyzed and brought together and integrated in artificial intelligence. So there's a lot of words about that. But in practice, it's just another tool, a new way of working with data, a new way of thinking. But it doesn't affect the experiments. Basically, the experiments are not affected by it. So what you do is you plan your experiments and you use the computer, the artificial intelligence to plan it and to integrate all the knowledge which is already available. So you make probably better experiments, but the experiment on its own is still similar as 10, 15 years ago.
And you can also use some of the computational biology methods such as single transcriptomics.
Yeah, but that's not computation. That is, again, a classical experiment, but large scale, where you get a large scale amount of data, but you have still to have cells or animals which you kill, you take the material out of it, and you analyze it with computer systems, data science. And that's, of course, what we do nowadays. Instead of one single experiment, where you analyze 1 or 2 parameters, you do now a big experiment and you analyze hundreds of parameters. So it's in fact a more effective use of the experiment.
Going back to your lab's use of animal models, you are also known for the human-mouse chimeric models. If you could just explain how these work, their advantages.
I think that's really explaining the dilemma when you discuss about animal experiments. So a mouse brain is 1 billion times more interesting as a study object than cell cultures of NAMs. As soon as you start to try to understand how a brain works, that's a mini computer if you compare it to the human brain, but it's at least a computer which works, which you can do things. The cell cultures are like you would take the computer down and you would put some electrical circuitries there, but you can't do anything beyond that. You can study parts of the electrical processes happening in a computer, but it's far from what. So the mouse is the cheapest, if I can say that, and the most easy way to approach a human brain. But it's a centimeter big and there is a lot of differences, obviously. But it's still a brain. So we worked for decades with this mouse brain, and we have really done a lot of things to understand Alzheimer's disease. But the problem is at a certain moment you are limited. So what we can do in the mouse is rather easily induce amyloid plaque formation. So the first steps, and we have this neuroinflammation. And what I was explaining earlier is largely based on that type of experiments. But then you want to go to the next steps, and that's the tau induction and neural death, but that doesn't happen in a mouse. So there must be something specific that makes that Alzheimer's disease is a human disease. And so in the end, it's always molecules which make the difference. So what we decide at a certain moment, and also influenced by the work of Pierre van der Hagen, who asks these questions about human-specific processes, but then during the brain development. And we thought we can maybe use the same approach to understand the human-specific elements in Alzheimer's disease. And so what we start to use is the IPS, the NAMs, the knowledge from the NAMs, to make neurons, human neurons, and we transplant those in a mouse context in a brain, where we induce the pathology, amyloid pathology, and these human neurons, in contrast to the mouse neurons next to them, these human neurons develop the full pathology. So the tangles, the tau, cell death, etc. And the same holds true for the other cells, the microglia, the inflammatory cells. So we know that a lot of the genetics has to do with how the microglia, how the inflammation is induced. So you do that in a mouse, you get the inflammation, but you don't get this further drive towards human pathology. So what we have done is we have basically, with certain treatments or genetically, you can remove all the microglia, the mouse microglia from the brain, and then we can use iPSC cells, stem cells, which we then treat in a certain way so that they become microglia precursors, as we call it. So that means cells which are capable to form microglia. And so we can inject them then in newborn pups which have no microglia, and we see that that's magic, of course. We don't know how it happens, but these microglia which we created in vitro start to repopulate the brain. And so if we want to understand the genetics of Alzheimer's disease, a fantastic example is a paper which we tried to publish, where we transplanted a pool of microglia. So microglia from person A, B, C, and D, and each of these persons has a different load of genetics. That's impossible to model in an animal. So you can do that in vitro, but then you don't know what you are studying. So what we did is, instead of transplanting one type of microglia, we used all microglia from all these different people and transplanted them together to see how these microglia react on the amyloid plaques. And so what we see is the genetic risk, polygenic risk, which we know that there are many, many small differences in your genetic makeup, which make certain people having a 10 to 100-fold higher risk for Alzheimer's than people who have not these things. But each mutation, each change gives you 2 or 3 or 4% increase. It's a combination which makes that you have a high increase of risk. And so by using cells from people with such a high risk and then people with a very low risk and transplanting them, we can follow how these different microglia react. Mm-hmm. And so we got a very interesting specific reaction. For those who want to know it, it's an HLA, there's an antigen presentation reaction. So these microglia react like they see a bacteria in the brain. And so we see that this reaction is directly correlated with the genetic risk that these patients have. So there's a fantastic breakthrough, and that's all done with these chimeric mice.
So you would say that your work, both here at the VIB-KU Leuven Center for Neuroscience at the UK Dementia Research Institute, has this very good combination of both human models and NAMs?
Absolutely. Good science, I mean, every model is a model. The real stuff is the human disease, is the person. So, of course, a mouse and a cell culture and an organoid is not giving you the full scale reality. So you need different models to approach that reality. And so model A gives you certain information, model B gives you other information, model C gives you another information, and you integrate that. And then you try also to integrate what we know from human. Because people say you need to do experiments in humans. Yeah, I mean, then we are talking about Nazi. So, but that's the real model. So when do you know enough to go to the real model? That's when you have a real therapeutic hypothesis. If you have sufficient evidence from all these different approaches that approach would help to cure Alzheimer's disease, then you can go to the real model. But before, The only thing you can do is descriptive. As I said in the beginning, what we knew about Alzheimer, let's say 20 years ago, was that there are plaques and tangles in the brain, but how they come there, whether they are important, whether they cause disease, whether they are a consequence or a cause, no, you can never ask that question in a human. And so that's something which many people, when they talk about experiments, don't understand. We don't want that. It's not needed that the mouse is the same as the human, because then if it was the same, it would be unethical. And in fact, I try to think about these discussions at a more abstract level. So if you find it unethical to use mouse to think about problems, yeah, and you say you need to use human organoids, yeah, when is such a human organoid going to be able to replace a mouse? That's when it forms a real mini brain. But now they describe it as mini brain, but just a pile of cells. But when it starts to be organized and when it starts to have electrophysiological activities which are similar to a normal brain. So when does it then, according to these people, when does it become unethical to do experiments on such a mini brain? So we need something which is a real brain. But when it becomes a real brain, yeah, then we have, I think, bigger ethical problems than when we use a mouse. Because in general, if you take a bit distance, using a mouse is killing a mouse, basically. I mean, if you kill a mouse in the context of science with a question, it's an ethical problem. If you kill a mouse at home, it's no problem. And if you kill a mouse in a restaurant, then it's the reverse, because you are obliged to do it. It's unethical not to kill it. So that shows to me that this discussion is not about the mouse, but it's about other principles. It's about whether you can do experiments, how far you can do experiments. It's an anti-science discussion. But okay, I hope I answered your question.
Yes, you did. And now I think to finish, what would you say are the next steps, especially in your research, to combine animal model research and NAMs for Alzheimer's disease?
Well, as I said, and there we come to themes which were discussed already. Before I was afraid to generate a lot of data, because the more data you get, the more difficult it becomes to understand. I mean, it's not because you describe hundreds of facts that you understand the problem. And so that was impossible in the past, hundreds of facts and then making some reasonable conclusions. So we asked very precise questions. But that has changed with the artificial intelligence, because you can generate a lot of data, you are not able to organize it, but together with the computer, you get structure in this data. So my view is that what we will have in the future is a kind of circle. So you will start to analyze data based on the knowledge, which is the general knowledge, the knowledge in your lab, etc. You will use your artificial intelligence to ask questions, and the machine will give you answers, will structure your information, and you will make a hypothesis, just like you did in the past. In the past, you had to read and think 3 months. Now you are sitting behind your computer, and after an hour, you have already the question precisely described. And so then you say, okay, how are we going to address this? What facts do we still need to fill that in? So again, with your computer, you will decide what are the best experiments. Then you will do a bulk experiment. In mouse, probably a lot of experiments will still be in animals, because the data can come from clinical data, but that's usually not precisely addressing the question, or that's usually already in your computer model that you try to make. And so so you will ask a couple of precise questions, and then you will do a couple of experiments. But you will, instead of doing one experiment and one data, you will do one big experiment, and you will get all the data out of it, and you will feed that in your system. And so the system will then help you to decide what of these observations are addressing the specific question you are doing. And so then you will refine your hypothesis again with the artificial intelligence, and then you will move it again to a next experiment, probably a therapeutic hypothesis in that case, where you have decided together with the machine, it's very likely if we modulate that molecule or that pathway, we will have a curative effect or for diseases. And then you test that hypothesis, but instead of doing again one, you will say what are the 10 different possibilities here, and you will test them all 10 in parallel. So instead of one mouse, I mean, instead of a group of 60 mice which you treat, to know treat and non-treat, you will take maybe hundreds of mice. You will do the 10 treatments, so the control group, and it's much more interesting because you can compare them all in parallel. And you will put all the data again in the computer to decide together with your artificial intelligence what's the right thing. So that's how I see my future.
Thank you so much.